topic by topic. We're going to have a look today at respiration. Respiration comes in two sorts, aerobic and anaerobic, and we have a look at basically how cells get their energy. And there are quite a few sort of ideas and experiments and things that we go along. Now, the first question is, I suppose, why do we need energy? Well, we need energy to basically move, or better word, contract the muscles. We need energy so that we can undergo active transport. If you remember, that is the movement of substances the wrong way. We are moving it from a low concentration to a high. We need it to build large molecules. We even need it to sometimes break down large molecules. We need it for things like cell division. This is not an extensive list, but it's basically the main reason that we need things. I suppose it also powers nerve cells. So, how does the cell get its energy? So, let's deal with that one. How? Well, it gets its energy generally from food, and in particular glucose. Now, not all food's going to be glucose, so we may need to convert that food into glucose or something similar. Now, once we've got this glucose, we change this into an energy carrying molecule. And this energy carrying molecule is, if you like, the way of thinking about it is the energy currency, and it's called a molecule of ATP, adenosine triphosphate. And this molecule stores all the energy, and that is used to do the movement, the active transport, building large molecules, breaking down large molecules, controlling cell division, controlling the nerve cells. All of that done by this little chemical ATP, and that's made from glucose. And that comes with the next question: where does this take place? And this is in a special place, and it is the mitochondrion. If you remember, with a cell, we've got our nucleus, we might have our endoplasmic reticulum with our ribosomes on it, but we've also got these small little items, the mitochondria. Now, we don't know, but we guess, we believe that a long time ago an ordinary little prokaryotic cell invaded another one. When it invaded this other one, they sort of struck up a symbiotic relationship. The one inside said, look, what I'll do is I'll give you abundant energy if you give me food and protection. And so something like a eukaryotic cell was created. So these cells have mitochondria in them, and this mitochondria is interesting because it contains all the enzymes for what we will call respiration, but it also contains its own DNA, which is like bacterial DNA. And so these things, they reproduce independently. So this little thing is the little powerhouse of the cell. So I suppose the next question we come to is, what is respiration? Well, respiration is where we take glucose plus we take oxygen and we make from this carbon dioxide plus water. Let's look at this equation. C6H12O6 plus 6 oxygens gives us 6 CO2, 6 carbon dioxides plus 6 water. It's easy to remember because it's all the sixes apart from that one, which is 12. So this is what I will call aerobic respiration. And we've got this molecule of ATP that does all the work. Well, I suppose we better have a look at this molecule ATP. I'm not going to go through in any great detail. For that you need to go on and do A-level, but we're going to try and just give some basic ideas about what it is and how it works. So what we have is a very clever system, and in the mitochondria we've got a little membrane, and on one side we've got lots of protons, and there's an enzyme which we'll call ATP synthase, and these hydrogen ions go through here, and as they go through here something magical happens. This bit turns, and what happens is a molecule of ADP comes in. I'll show you what that looks like in a moment, plus phosphate, and as it goes in, as it's turned, these two are pushed together and out pops out ATP. So let's have a look and see what this looks like. I've got a molecule of adenosine. Now adenosine, you'll come across, it's found in DNA, and attached to this I've got two phosphate molecules. And what we do is we take along another phosphate molecule plus water, and what happens, we get here and we get an extra phosphate put on. Now this bond is basically very weak. We need water here to make it work. But this bond is very weak, and at a moment's notice that can be lost, and as it's lost, energy is released. So we go back that way and we get this energy released. So we've got this simple chemical just storing the energy, and you don't have too many molecules of that because they are constantly being reused. So you make your energy, use it, and then you remake it, this energy store. So this is called our energy currency. So this is our basic idea, that we've got mitochondria powering the cell, but other parts of the cell may do quite good bits and pieces. So what we have for this idea of respiration is we take something like glucose. We in fact add some ATP to this, and what we do is we actually make glucose with some phosphates joined to it, and then this glucose breaks down into a chemical called pyruvate, can't spell, and that can go then one of three ways. We can either take it through what we'll call anaerobic respiration, and this is done differently in plants and in animals, or we just go down and we do aerobic respiration, where this is basically broken down into carbon dioxide and water. In animals we make lactate, or sometimes you'll see this referred to as lactic acid. Take your pick, and then that is basically later converted back via aerobic respiration, and this you'll see called paying the oxygen debt. And the other one that we've got are plants, and what they do is they break this down into carbon dioxide and ethanol, and it's also done by the yeasts, and this is really incredibly useful to us because this way we can make bread and alcohol. So here is our basic idea of respiration. Now, what are we going to try and do with this? Well, there are a lot of experiments that we can do, but many of them take a long time to actually do. And I'm going to talk about some of those and give you some ideas about them, but I can't do many of them here because they're going to take too long, and for that you've got to watch some of my other films. So let's have a look at some ideas. Right, the first of these is we're going to meet and use this substance, lime water. I've made some lime water here. I've taken some calcium hydroxide, put it in water, shaken it up, left it. You can see there's a white powder at the bottom, and what I do with that is we simply filter this, and there's my nice clear lime water. So I'll leave that to cook. Let's have a look at some experiments, and we're going to have a look at this particular bottle. When I ask many students to do this experiment, what I will do is give them a much simpler system, and what I can give them is a boiling tube and a straw, and I'd say to them, take this, blow in there, and usually it goes all over them. So instead, this is a special container, and if we put air in here, it goes down right into this tube and it goes into this liquid, lime water, bubbles up and then comes out through this tube. So let's have a look at this and see what happens when I blow into it. Well, not a lot happening here at the moment. My experiments don't seem to be working well at the moment. There's lime water freshly made up and it's not really going very cloudy. That's a bit of a failure, that. Let's try a bit more. I've got some fresh lime water. This is the other way of trying to do it. You have to blow gently. Looks like my lime water's not working. Right. How boring. Right. There are other ways of testing this, and one of the other ways of testing this is to use a different chemical, and this is bicarbonate indicator, and this will try, it's a bit different. You'll see it sort of collapsed the bottle where it's absorbed any gas that was in there. And what we're going to do is going to put a little bit of bicarbonate indicator in there, and let's try blowing in. Let's see if this one works. What I'm looking for here is a colour change. Well, can you see any colour change there? Not much. Right, I think I'll give up experiments. It doesn't seem to be working at all well. Can I just have some tissue? There we go. Right. So, lime water should go cloudy, and it's not. That must be something to do with what I'm trying to do, and could suggest that I'm dead, of course. Well, that didn't work quite as planned, so we'll move on. Now, one of the things that we're asked to think about when we try and do this is, can you design an experiment to work out basically about living organisms and the fact that they produce carbon dioxide? Let's have a look at some ideas here. So we've got to try and design an experiment. Designing an experiment's not going to be particularly easy, and so let's take one of those boiling tubes that I had and see if we can come up with an experiment. And what we can do is we can place at the bottom some of this bicarbonate indicator. Now this is in theory sensitive to small changes in carbon dioxide. So what we can sometimes do is take a piece of then wire gauze and we might be able to put some sort of insect there, and then we could stopper the bottle for a little while. Hopefully they would take glucose plus oxygen in the air there and go along and produce carbon dioxide and water, and that carbon dioxide will change the bicarbonate indicator and you will see a colour change. And we can see that my bicarbonate indicator didn't seem to work, and we can also see that the lime water didn't. The lime water wouldn't work with this because you'd need to bubble it through, and for some reason mine's not. So that's a way of trying to look at small organisms. Alternatively, here you could replace this perhaps with a plant or seeds. Probably a bit more ethical to do the seeds. We've got to make sure that when you put the insects in there that you can get them out and you can release them and you don't kill them, because it's rather cruel doing that. So there is this one to show that carbon dioxide is produced by small organisms. A different experiment that you might come up with is basically to take a flask, and by this I do mean a proper flask, so we've got our vacuum flask, we'll put in here a thermometer, seal it, and then we'll surround this here with germinating something like peas. The way we would go about this is you take some dried peas, you'd soak them for 24 hours, start getting them to germinate. Then you'd set up your vacuum flask with the germinating seeds in there. Now what you will need to do is make sure that before we do this we actually clean the seeds, and once we've done that, to set up the vacuum flask, first of all, what I would do is I would rinse the peas. 2, I'm going to wash them in about 1% bleach. Then I'm going to rinse the peas again, and then I'm going to rinse them again, and then place in the flask plus the thermometer, plus a lid. And as you can see here, this is all done upside down, and we've done that to make sure that this thermometer is inside touching the peas. And then this thermometer, what I would do is I'd have a digital thermometer connected to my computer, and then here we would start the computer, tracking, and what we should see then is over a period of time the temperature will go up, looking something like this. Temperature and time. And we should see that the peas will rise in temperature maybe from something like 20 to 23 Celsius. It won't be very much, but it gives us an idea that heat or energy is basically given out. So heat is produced by respiration. And you know that simply because if we leave you just standing, or we have you running, then when you get running, basically you get hot. And this is how we can sort of measure respiration, that we actually do get rather hot when we're trying to do this. But normally you're going to get hot, and we use the heat from respiration to keep the body temperature at 37 Celsius. So we've got this idea of respiration and the need for respiration. When I gave the list before, one of the things I didn't put down is simply heat. We need energy to keep basically warm. We keep here and maintain a constant body temperature, and this is typically just for mammals and birds. So we could keep this constant by simply either cooling, but to warm up we need respiration. Cooling, you can use things like sweating, which is all about evaporation. But warm, you can only stay warm by respiration. So we've got the major functions of living cells, and some organisms are going to need to do this one. So there is our basic need for respiration. We've got this idea of living cells needing energy to carry out their basic forms of life and to move around. So let's now look at the response to exercise. Now, to try and do this, what we've got to do is look at things like breathing rate. And there are a whole series of different ways we can actually do this. We can look at perhaps the breathing rate as first. We can look at the number of times you're breathing per minute. You can look at other things like the blood circulation. You can look at the heartbeat to see how many times that's going to actually go up. We can measure the amount of carbon dioxide given off while we're doing different sort of exercises and the like. So a normal type of experiment here is going to take someone that's resting. And while they're resting, what we can do is we can measure various things. So what sort of things could we measure? Well, we can measure the heart rate. Now, how would you do this? Well, there are a whole series of different things you can use. The obvious one is one of those things that you put on your finger. I've put it out on the side. I forgot to bring it in. So we can measure your heart rate, and these things also measure the O2 saturation. And here is one of those things. Now, the only people it doesn't work well on are young ladies who've got painted nails, but otherwise we just stick this on and we can start this. And I have a problem trying to show you this because simply my fingers are around the wrong way. So I've got here my pulse being taken, and it tells you my heart rate is 73, and if we can put this round that way, just about to see. I'll zoom out just a little bit and you can see that my sats are 96. So these little pulse oximeters are fairly easy to sort of use, and we can measure simply our heart rate and our O2 saturation. We can then go off and do some exercise. And How long you're going to do that depends on how fit you are, but this could be anything from 1 minute to sort of 10 minutes of exercise. That's depend on what you can do. And we can measure the heart rate again, and you can measure the O2. So we should see that changing. We can look at other things using special machines, and one of those machines is a spirometer. And the spirometer, there are a variety of different types, but basically you breathe into a machine and you breathe out, and it can simply measure the amount that you breathe and also the rate at which you breathe. But neither of these things are actually directly measuring respiration. What they're looking at are other things. So let's have a look at a little graph of this. So let's come up here with heart rate, and on the bottom we're going to come up with time. So this is my period of rest. My heart rate is low. Then we're going to start some exercise at this point here, and what we see is my heart rate goes up very rapidly, and then stays and then when I come to do some rest, it's going to return to normal. If I, instead of doing this, I look at my breathing rate, and we'll do this one in red, my breathing rate is going to be fairly constant. Then my breathing rate's going to slowly increase, and then when I start resting, it's going to come back down to where it was beginning. So we've got an idea of what we're doing here. What we do see, however, is there is a real difference between someone that is fit and someone who is unfit. So the sort of things that we're going to look at is the amount of blood pumped. So if we look at the amount of blood pumped, this is, if you like, per beat, a fit person might do something like about 65, and an unfit person might do something like about 80. We might look for the volume of a breath, and the volume for a breath for a fit person might be 120, whereas an unfit person might be 140. We might look at the resting breathing rate. And this person might have a higher rate, 15, compared with the other person at 12. And we might have things like a resting pulse rate, and a fit person might have a higher pulse rate than the unfit person. Or could it be? You see, let's have a look. Does that make sense? The fit person has more blood, so this will be our fit person, and this will be our unfit. They've got this higher rate. Simply, they can do more blood per beat. They can breathe basically more air per breath. Because they can move more blood per beat, their heart beats slower. Therefore, they need to push less blood round.
Here's your reminder. Take imperatin tablets.
So, we have to think about these. We have to think quite carefully what we've got. What would appear to be an unfit and fit person, they're round the other way. The unfit person here look better, but they weren't. And it's these clues that give things away, whether we're looking at a fit or unfit person. So, to be fit, what have you got to have? You've got to have the ability to here convert glucose to energy at a reasonable rate, and as well as that, you've got to be able to pump blood around the system efficiently and to breathe and move sort of a fair amount of oxygen. So our breathing rate usually involves having things like larger lungs so that we can actually breathe in more. And this is where we see a slight difference between men and women, and this is a difference really in anatomy. Generally men are larger chests than women, so they've generally got larger lung capacity. Generally. Doesn't mean to say they need it in all cases. So we've looked here at aerobic respiration, and next time we need to have a look at anaerobic respiration, and we'll have a look at some other bits and pieces to do with metabolism. So until then, if you found this useful, then please subscribe, and I'll see you next time on GCSE Biology, topic by topic. Till then, bye bye.